Hey everyone, my name is Antonio Mendez, and I'm going to be presenting my research-based capstone in this Inspire Talk format. Today, I'll be discussing a protein called CROS, and by the end of this talk, my goal is to explain the relationships between CROS, DNA polymerase 5, whiteout, antibiotic resistance, E. coli, and DNA damage. And you can see here in the center of the slide, I've sort of represented this with a GIF of Charles Darwin boxing DNA. So DNA damage can arise from endogenous processes, which are derived from within a cell. And these can include deamination events, base substitutions, strand slippage, and depurination reactions. But damage can also arise from exogenous agents, and these originate from a cell's environment rather than from cellular processes, and include things like UV radiation or treatment with drugs like fluoroquinolones, which are antibiotics that target DNA replication enzymes. So after one of these DNA damaging events occurs in E. coli, which is the bug we use in our research, lesions are left behind. For example, at the top of this panel, we can see a thymine-thymine dimer, which is generated by UV radiation, and it's causing DNA polymerase 3 to stall because it can't elongate past this lesion. As a result, the replication apparatus disassembles and reinitiates downstream like we see at the bottom, and this leaves behind single-stranded DNA, which inhibits the essential processes of replication and transcription. So to survive, E. coli has evolved DNA repair mechanisms, which are collectively known as the SOS response. This response is temporal in that the first mechanisms deployed are error-free, and these processes are sort of like whiteout because the cell can cut out and replace DNA lesions, or white them out to write down the correct sequence. However, if these strategies fail, the error-prone processes are activated, which involve the activity of DNA polymerase 5. The SOS response is induced after a protein called RECA, which is represented by the pink blob at the top, binds to single-stranded DNA, which we know is left behind following DNA damage. This forms what we call activated RECA, and it's activated because it stimulates autocleavage of the LEX-A protein, which represses the SOS response, like we see on the left. LEX-A cleavage, which we see on the right, then allows SOS response genes to be expressed. If damage persists after SOS induction, the last resort of this response is expression of DNA polymerase 5, and this is because Pol5 can bypass DNA lesions in a process called TLS, but this process is error-prone and leaves behind mutations. Our research involves an ortholog of Pol5 that is encoded by the RUMA-B genes, which are carried on the highly mobile genetic element ICE391. And this mobility means that these genes can be easily transferred between bacteria. Now, Pol5 expression is tightly regulated to avoid unnecessary mutations, or mutagenesis, because these are typically bad for a cell. An example of this includes LEX-A repression. Interestingly, very low levels of mutagenesis are promoted by ICE391, which encodes Pol5 in addition to dozens of other genes. But mutagenesis increases robustly if we take away these other genes and only express Pol5. This suggests that genes on ICE391 downregulate Pol5 expression, and we identified CROS as one of them. So to test CROS activity, we perform mutagenesis assays, which are really simple because high mutagenesis means elevated expression of Pol5 and low mutagenesis means low expression of Pol5. Now, if we look at the center bar on this graph, we see low mutagenesis promoted by co-expression of Pol5 and CROS. However, if we take away CROS, like we see on the rightmost bar, high levels of mutagenesis are promoted, and this seems to confirm that CROS is a negative regulator of Pol5. And we can see the same trend when we monitor the effects of CROS on the amount of Pol5 proteins generated from Pol5 expression. Here, we are measuring the cellular quantity of one of these proteins, called RUMA, which corresponds to the band intensity on this image. In the presence of CROS, very low levels of RUMA are detected, as indicated by the faint band on the left. But if we look to the right, we see a very intense band, corresponding to high levels of RUMA when CROS is absent. So we have identified CROS as a factor that attenuates the activity of the DNA polymerase 5 ortholog encoded on ICE391. The mutagenesis promoted by Pol5 contributes to the de novo generation of virulence factors in bacteria, like antibiotic resistance. Due to the mobility of ICE391, this mutagenic polymerase can be easily transmitted between bacteria, augmenting the threat of multi-drug-resistant pathogens. The first step in combating this is understanding how these virulent phenotypes manifest, which is what we have done here.